Okay, this is SYJC biology lecture, chapter number 1. Name of the chapter is Reproduction in Lower and Higher Plants. We are in part 3 of this chapter. I am Professor Prasad Tamankar. Let us proceed with the pollen pistil interaction that we had come up to. Okay, so last time we did something like regarding pollination process. Pollination, different aspects we have done. Within that different aspects, we first consider what is pollination, the definition, the types of pollination, auto-pollination or self-pollination and cross-pollination. Self-pollination with a autogamy and geitonogamy, cross-pollination with a xenogamy and hybridization. Then we understood the different agencies for pollination. Okay, and at the end, agencies we did two types of agencies. One type of agency was biotic type of agencies and other was abiotic. Abiotic में था anemophily and hydrophily. Hydrophily was of two types, underwater and on the surface. Underwater is called as hypohydrophily. On the surface it is called as epihydrophily. Then we studied the biotic agencies. In that case, we understood about the entomophily. Then we understood milacophily, then chiropterophily, and even ornithophily. So the pollination which is controlled by insects, ornithophily means birds, chiropterophily means bats, and milacophily means snails. So different agencies are there which are carrying out process of pollination that we understood. Then we studied the contrivances for pollination. Contrivances means ideas. So contrivances for self-pollination and contrivances for cross-pollination. Contrivances for cross-pollination were basically to have cross-pollination done first. And in that case, it was also called as outbreeding devices because cross-pollination is also called as outbreeding. Self-pollination is called as inbreeding. So inbreeding में क्या है? Bisexuality है, then cleistogamy and underground flowers. Okay. Then in case of cross-pollination, we had some ideas like unisexuality. Flowers are unisexual or they're present separately on same plant. Means male flowers and female flowers are present on same plant at different position, or otherwise the plants are different totally from each other. That is unisexuality. After unisexuality, we understood hercogamy. Hercogamy was a difference in maturation time. So prototype, protogyny we understood. Then there was a process of heterostyly. Length of style will be different with respect to length of what was that? Stigma, okay, or filament, let us say. That was one idea. And self-sterilization. So ये कुछ ideas थे. Now we have to understand the things ahead. We will be going for fertilization. How fertilization takes place, what type of fertilization it is, किस तरह से ये होता है, that all we are going to understand. So we need to go and study in that particular respect. So fertilization करते वक्त अपने को पहला point करना है, that is pollen pistil interaction. Now what is pollen? Pollen is microspore. Microspore is produced in anther. Anther gets dehisced, means broken, and pollen grains are distributed all over the surrounding, and agencies, proper type of agency will carry it from anther to the stigma. So stigma actually is a receiving end of pollen grain or microspore. Remember, pistil actually is a unit of gynoecium. This pistil will consist of ovary, style and stigma. So stigma is the receiving end जहाँ पे pollen grain आके गिर जाता है और यहाँ से शुरू होता है उनका interaction. So let us continue with the story. Okay, it is an interaction between pollen grain, that is microspore, and stigma, that is sporophytic tissue of plants. Then all the leaves from deposition of pollen grains, all where we can say deposition of pollen grains on stigma to the entry of pollen tube will be considered as pollen pistil interaction. मतलब सिर्फ pollen grain आके गिरेगा नहीं, वहां पे वो germinate होगा, pollen tube तैयार होगा, pollen tube will be made to pass through style, it will reach up to ovule and will try to enter into the ovule. So all these are together considered as pollen pistil interaction. Okay. Now pollination is a chance factor. Chance factor का मतलब you know very well. If the flowers are cross pollination and if flowers are different distant places, then reaching pollen grain to a particular stigma may be or may not be, means it is a chance factor. हो सकता है या नहीं भी हो सकता है, ऑफ कोर्स इट हैपेंस मेनी अ टाइम्स, बट चांस है, एक पोलिनेशन होना या ना होना चांस है. ओके, सो ड्यूरिंग दिस पर्टिकुलर थिंग some wrong pollen grains or unwanted pollen grains also might be brought to the stigma. ऐसा जरूरी नहीं बनता है कि भिंडी का फ्लावर है तो भिंडी के फ्लावर के ऊपर भिंडी का पोलन ग्रेन ही आके गिरेगा. ऐसे हो सकता है कि नारियल मतलब कोकोनट के प्लांट का पोलन भी आके गिर सकता है, ज्वार प्लांट का पोलन ग्रेन भी आके गिर सकता है. बट द पिस्टिल is capable of recognizing the suitable type of pollen grains, and they are capable of understanding incompatible or non-compatible type of uh, pollen grains. So compatible and non-compatible type of pollen grains would be understood by the pistil, and the compatible one will be allowed to germinate. नॉन कंपेटिबल जो पोलन ग्रेन्स होंगे, मतलब जो अपने स्पीशीज के नहीं है या फिर अपने टाइप के नहीं है, they will not be allowed to germinate. The rest, the one which are of same type, same species, will be allowed to germinate, and then the germination process will begin. Stigma over here will secrete some sugary watery solution. This sugary watery solution will be absorbed by the pollen or pollen grain, we can say. Then it will start growing. So this nutrition will be provided by स्टिग्मा, बिकॉज पोलन के पास कुछ भी नहीं है. पोलन ग्रेन तो ऐसे ही शेड किया जाएगा या छोड़ दिया जाएगा हवा के अंदर, या मे बी ऑन एंथर, इट विल बी देन कैरिड टू स्टिग्मा. ओके, ओनली सम इंट्रास्पेसिफिक पोलन ग्रेन्स आर अलाउड टू ग्रो. इंट्रास्पेसिफिक मतलब समझ में आया ना, अपने स्पीशीज के या क्लोजली रिलेटेड स्पीशीज अगर होगा तो ये पोलन ग्रेन्स को जर्मिनेट हुआ या होने दिया जाता है, अदरवाइज दे आर नॉट अलाउड टू. ओके, characteristic feature. Fine. This is one more important characteristic feature. The pollen tube which is developed, this pollen tube may travel through style. Okay. स्टिग्मा को क्रॉस करेगा, पियर्स करेगा, मतलब छेद करेगा स्टिग्मा को. It will enter into style and will be pushed into ovule very close to embryo sac. अभी आपको पता होगा कि ओव्यूल के अंदर एम्ब्रियो सैक कहाँ पे होता है, उसके अंदर एग apparatus जहाँ पे है उसके पास ये पोलन ट्यूब पहुंचना चाहिए, and then the male gametes in it are released. So this is the general mechanism that is followed. Okay, so remember it very well. Here we are trying to show this kind of thing. The first important thing, like say this is a entire pistil available. Pistil के ऊपर ये जो part है इसको हम लोग कहेंगे ये entire pistil है. तो इसमें ovary के अंदर ये ovule है, and you could see the ovule has got matured. Means inside you can find over here egg apparatus, antipodal cells, then secondary nucleus are present. Then this particular part is style, and this is stigma. So onto the stigma there are pollen grains already shaded. We could say that there are different types of pollen grains shaded. So to indicate the pollen grains are of different type, they are named or numbered like S1, S2, S3, S4, etc. Okay, so suitable pollen grains are allowed to germinate. So S2, S4 और S3, S4 are allowed to germinate. यहाँ पे S3 और S1 है तो S1 को जर्मिनेट नहीं किया जाएगा, S3 को जर्मिनेट, S2, S1 को जर्मिनेट नहीं किया जाएगा at all. So this is how it will happen. So suitable pollen grain will be capable of germinating. Pollen grain will absorb water as well as nutrients, as I told you, as a result of which germinating pollen grains will start producing a long tube-like structure. This is called as pollen tube. Pollen tube then will reach up to what? Up to the ovule, and will try to enter into the ovule. Here it will try to come very close to the ovule and to the synergid cells. They will absorb what? Water from synergid cells. Synergid cells को कहते हैं filiform apparatus, है ना? ये भी हम लोग ने last time देखा था. So as a result, it absorbs water from this filiform apparatus, that is synergid cells. The pollen tube will become turgid, and then it ruptures. When it ruptures, the pollen grains, the pollen tube that contains male gametes are released inside, and these male gametes will carry out the process of fertilization. Okay, so this is all in all considered as pollen pistil interaction. Now, what is significance of this? 2 points we have to mention. Developing competition among the compatible pollen grains. जो healthy pollen grain होगा, compatible होगा, that only will be allowed to germinate. Rest are not. So the future generation will become more strong or more suitable, more adaptable, and it plays a very important role in sexual reproduction and seed formation. It is responsible for carrying out variations in the next generation, so that what will happen, there will be the powerful next generation or good next generation. So that's all we can say about the pollen pistil mechanism. Now, germination of pollen grain can be stimulated in vitro. ये एक्सपेरिमेंट आपको करने का है. बायोलॉजी लैब में जब जाओगे शायद ये एक्सपेरिमेंट आपको रहेगा. मैंने पहले भी एक बार कहा था शायद कि you will be provided with China rose flower which is blossomed. Then you will see the pollen grains are there released from anther. What you are supposed to do is take a slide, put a drop of glycerin or water, maybe whatever it is, and then tap the pollen grains or tap the androecium part. Okay, it is a monodelphous stamen found in China rose. So you have to tap that particular thing. Pollen grains will shed in the liquid, put the cover slip, wait for some 10-20 minutes, and you could see that the pollen grain will germinate so as to develop a pollen tube. You have to display it under microscope and show it to examiner. That is a simple experiment. Okay, so remember this very well. For germination, some factors are essential, like sucrose is essential and boric acid are useful. So these two, if are present, they will be stimulating the germination of pollen grain and accelerate the pollen grain germination so as to form the pollen tube. Okay, तो आपको experiment करना है तो experiment भी कर सकते हो. Take only water, means plain water, add sucrose to water and make that, and add sucrose and boric acid. बोरिक एसिड मतलब आपको मालूम होगा ना, कैरम बोर्ड खेलने के लिए हम लोग जो पाउडर डालते हैं कैरम बोर्ड के ऊपर उसको ही बोरिक एसिड कहते हैं. ठीक है, तो ये आपको आराम से घर में मिल सकता है. ठीक है, तो जनरली हम लोग ये यूज करते हैं, चावल को उसको मल के रख देते हैं, चावल को अप्लाई करके रख देते हैं बोरिक एसिड, सो दैट देयर विल नॉट बी एनी पेस्ट्स इन इट. सब कुछ कीड़े वीड़े नहीं हो, तो मम्मी ये यूज करती रहेगी घर में अलग पर्पस के लिए. आप लोग कैरम बोर्ड खेलते वक्त उसको यूज करते रहेंगे. ओके, now there is one part that is called as artificial hybridization. What is artificial hybridization? It is transfer of pollen grains from a desired anther to the stigma of desired type. मतलब ये दोनों specific होगा. Where it is done or why it is done? This is done so as to carry out some hybridization experiments. Hybridization experiments करवाने के लिए इसको किया जाएगा as Mendel did it for carrying out some specific type of breeding. है ना, उनको चाहिए था कि दो अलग अलग कैरेक्टर्स को मिक्स करना था. So in that context he did it. Even today it is done. Okay, and it is carried out so as to have a specific hybridization done. So remember this very well. Pollen grains are collected. Okay, अभी pollen grains को collect करना पड़ेगा and then they have to be transferred to the stigma with the help of smooth brush. अपने जो हम लोग पेंटिंग करने के लिए जो ब्रश यूज करते हैं, पेंटिंग मतलब वॉल पेंटिंग नहीं, ड्राइंग वाला पेंटिंग जो यूज करते हैं, उसके छोटे छोटे ब्रशेस होते हैं, उसको हम लोग यूज कर सकते हैं. These brushes are soft, और sometimes if the flowers are larger then sponges are also used. Okay, thin पतला सा स्पॉन्ज लिया जाएगा, उसको थोड़ा सा गीला कर दिया जाएगा, पोलन ग्रेन्स के अंदर डुबो के फूल के ऊपर, जैसे कि सनफ्लावर उसके ऊपर ये कर सकते हैं so as to increase the product of sunflowers. Why? Because these oil seeds, sunflower is giving us oil seed for sunflower oil, and so उसके लिए ज्यादा अगर pollination होगा तो ज्यादा oil seeds मिलेंगे. So for that purpose even it is done. So this is a general characteristic feature. The technique will involve emasculation and bagging. अभी ये थोड़ा सा हम लोग अलग चैप्टर में करेंगे. What is this? Emasculation. कभी कभी flowers जो होते हैं, flowers are bisexual. These bisexual flowers need to be converted into unisexual male flowers. Is it? No. Unisexual female flowers. ठीक है. Female flowers क्यों? जिसको हमें पोलन ग्रेन्स डालने हैं, शेड करने हैं, वो फ्लावर में सिर्फ फीमेल पार्ट होना चाहिए. हाइब्रिडाइजेशन कर रहे हैं हम लोग, लाइक ए एंड बी. सो ए इज टॉल, बी इज ड्वार्फ, उसके बीच में हाइब्रिडाइजेशन करना है अपने को. सो टॉल प्लांट के पोलन ग्रेन्स अपने को ड्वार्फ फ्लावर के ऊपर डालने हैं, ड्वार्फ प्लांट के फ्लावर के ऊपर डालने हैं. बट बोथ आर बाइसेक्सुअल प्लांट्स. सो व्हाट वी शैल डू? द बी प्लांट विच इज रिसीविंग, विच वी आर कंसिडरिंग एज फीमेल प्लांट, वो तो बाइसेक्सुअल एक्चुअली, लेकिन उसको फीमेल में कन्वर्ट करना पड़ेगा. उसके लिए फॉर दैट पर्पस व्हाट इज डन, द anthers are emasculated, means anthers are removed, plucked, or maybe some treatment is given so that the anthers will become neuter, or anthers will not be there, and means male part will not be there, so female part only will remain. So this bisexual flower will be converted into female flower. That is the logic behind emasculation. Now after emasculation, the emasculated flower should be bagged. Bagged का मतलब क्या? उसके ऊपर एक टिश्यू पेपर और बटर पेपर का बैग डाल सकते हैं, प्लास्टिक का बैग डाल सकते हैं. And then it is tied. Why this is tied? Because we are interested in performing desired type of cross pollination, desired type of pollen transfer. अगर हम लोग उसको ऐसे खुला ही छोड़ देंगे तो क्या हो जाएगा? Any insect or any other media will be capable of carrying out pollination of this emasculated flower. तो अपना experiment पूरा बिगड़ जाएगा. And that's why emasculation करने के बाद bagging is essential, and then we will wait for a suitable cross that is carried out, and then the product will be obtained. Now in this case we are going to understand the double fertilization. अभी ये सब जो हम लोगों ने कहा hybridization technique, उसका थोड़ा सा पार्ट हम लोग दूसरे एक चैप्टर में देखने वाले हैं. वहां पे मैं और एक डिटेल में एक्सप्लेन करूंगा, क्योंकि emasculation के अलग अलग टाइप्स है. Emasculation can be done by plucking method, by scissor method, by hot water treatment method और by अल्कोहल ट्रीटमेंट मेथड. वो हम लोग बाद में डिस्कस करेंगे, अभी नहीं डिस्कस करेंगे. वी आर करंटली गोइंग फॉर डबल फर्टिलाइजेशन, विच इज अ कैरेक्टरिस्टिक फीचर ऑफ एंजियोस्पर्म. Namaschin tried to find it out using lilium and fritillary, okay, type of plants को यूज करके उन्होंने उसको यूज किया. Now what is this double fertilization and how it happens? We have to start a story right again from pollination. Okay, so why it is called as double fertilization? उसका definition पहले देख लो. Double fertilization is so called because fertilization takes place twice. Okay, there are two male gametes present in pollen tube. Remember it very well. A male gametophyte in case of angiosperms produces two male gametes. Out of two male gametes, one male gamete will fuse with egg cell and other will fuse with secondary nucleus, and fertilization will be carried out. As it is occurring two times, it will be called as double fertilization. As I told you earlier, it is a typical characteristic feature of angiosperms. Angiosperms का property है ये, so will be carried out only in case of angiosperms, nowhere else. In case of gymnosperms, there are two male gametes produced, but the two male gametes, though they are produced by gymnosperms, only one will be utilized. The second one will degenerate. Okay, in case of angiosperm, second male gamete is utilized for formation of endosperm tissue. ऐसा वाला चीज gymnosperms में नहीं होता है, and that's why it is a characteristic feature of only angiosperms. Remember it well. As I told you, one male gamete will fuse with egg cell to form zygote, and the other male gamete will unite with secondary nucleus so as to form tertiary nucleus or pen nucleus. और pen का मतलब ही primary endosperm nucleus होता है. So ये दो चीजें तैयार हो जाएंगी. After pollination is done, the pollen grain will germinate first. That we have said already onto stigma. ये germination हो जाएगा. And when germination is completed or completely done, what will happen next? The germination will start forming a pollen tube. That pollen tube will be made to pass through stigma. Okay, देखो यहाँ पे pollination हो रहा है. Pollination, there are many pollen grains, each one is undergoing germination, and germination के बाद ये लोग pollen tube बना रहे. The one which is making pollen tube will be made to pass through this part. इस part को हम लोग कहेंगे, क्या कहेंगे इसको? Style कहेंगे. So पहले stigma के पास through pass हो जाएगा, then it will be made to pass through style and will reach up to the ovule. This is a general feature, so as to understand. Okay, so remember this very well. ये पहला पार्ट है. Now journey of pollen tube will be guided by some chemicals. Chemicals are secreted by synergid cells. Okay, this kind of movement will be called as chemotactic movement. Okay, generally it is a malic acid. Malic acid is being secreted. Malic acid is sweet to taste. So generally most of the plants make use of this malic acid as a what we can say bait. Towards this smell of malic acid, the male gametes or maybe the pollen tube, they will start progressing and they will try to carry out the process of fertilization. Now उसमें बहुत सारे stages involved है. Those stages we are going to study. First stage is germination, formation of pollen tube. How pollen tube passes through style again is a matter of question. वो बाद में अगर बताऊंगा मैं, अगर इसमें नहीं आएगा तो बाद में बताऊंगा. Okay, first यहाँ पे होगा penetration of ovule by pollen tube. That will be done first. Then there will be porogamy. It is porogamy is a kind of event where pollen tube will try to enter into ovule through micropyle. Okay, तो पहले मैं थोड़ा सा एक्सप्लेन करूँ दूसरा, उसके बाद में इस इवेंट पे आते हैं. सो अगर हम लोग डबल फर्टिलाइजेशन का पूरा इवेंट देखना चाहेंगे तो पहला इवेंट है पॉलिनेशन, दूसरा इवेंट है पोलन पिस्टिल इंटरेक्शन, तीसरा इवेंट है जर्मिनेशन. जर्मिनेशन में पोलन ट्यूब तैयार हो जाता है, यू नो वेरी वेल. फोर्थ इज द पासेज ऑफ पोलन ट्यूब थ्रू स्टाइल. अभी स्टाइल के थ्रू कैसे पास होगा पोलन ट्यूब, दैट इज इंपॉर्टेंट. ठीक है, देखो ये थोड़ा सा नीट पॉइंट ऑफ व्यू से मैं बता रहा हूँ, अपने बुक में शायद ओमिट किया हुआ है, दैट्स व्हाई इट इज नॉट देयर इन आवर PowerPoint also. So I'm explaining it to you. Now see, the style may be hollow in some cases or style may be solid. Style is hollow means what, उसके अंदर lumen होगा. So for hollow style, pollen tube will make use of side wall and will take support of side wall and will pass in. Okay, if pollen tube, if the style is spongy means solid, then pollen tube will pass through intercellular spaces. Then pollen tube will pass through intercellular spaces. So there are two ways in which the pollen tube penetrates through style. कौन से दो थे? You must have got it. If for hollow style, pollen tube will take support of the side wall of or inner wall of style, and if style is spongy, the pollen tube will take support of intercellular spaces of cells of style and will pass through, and then ultimately it will penetrate, will reach close to the ovule and will now try to enter into the ovule. This entry of pollen tube into ovule will be considered as penetration of ovule by pollen tube. There are three ways. Okay, number one is, as it is shown over here, porogamy. What is porogamy? Pore का मतलब होता है aperture. See, there is a small aperture left. This is called as micropyle. So pollen tube will come very close to micropyle and will try to enter in to the micropyle. That will be called as porogamy type of event. Second event will be there called as chalazogamy. What is chalazogamy? In case of chalazogamy, what will happen? The pollen tube will enter in, will reach over here, will try to enter in through chalazal end. Chalazal end के थ्रू ही अंदर आने का कोशिश करेगा. देखो यहाँ से ये अंदर हो चुका है, बट अल्टीमेटली इट हैज टू रीच अप टू हियर. इधर क्या हुआ, पोलन ट्यूब के ओव्यूल के बाहर से ये आके पोर्स के अंदर गया. यहाँ पे क्या हुआ, इधर से अंदर घुसा है. ओके, चलाजल एंड जो है ओव्यूल का, थ्रू दिस चलाजल एंड इट विल ट्राई टू पेनिट्रेट, एंड दैट्स व्हाट इज कॉल्ड एज चलाजोगामी. Sometimes it will try to penetrate the ovule somewhere between chalaza and micropyle. It will be then called as mesogamy. Okay, mesogamy क्यों कहेंगे? Because middle में देखो अंदर, यहाँ पे बीच में से कहीं पे अंदर आ रहा है, अल्टीमेटली उसको यहीं पे आना है. Ultimately it has to reach up to egg apparatus. But there are three ways. तो ये 3 मार्क का क्वेश्चन हो गया. What is penetration of ovule by pollen tube, and give the 3 different types studied by you. Okay, so what you have to say, entry of pollen tube into ovule will be considered as, entry of pollen tube into ovule will be considered as penetration of ovule by pollen tube. There are 3 ways: chalazogamy, porogamy, mesogamy. In chalazogamy, the pollen tube is going to penetrate from chalazal end. In porogamy, it is try to enter through micropylar end, and in mesogamy, pollen tube will enter into ovule somewhere in between chalazal end and micropylar end. So ultimately it will reach close to the egg apparatus. कहीं से भी अंदर आए, ovule में कहीं से भी घुसे, ultimately it has to reach very close to the egg apparatus. Egg apparatus has got synergid cells present over there. The pollen tube will absorb now water from synergid cells. Synergid cells में से पानी लेने की वजह से क्या हो जाएगा, ये थोड़ा सा turgid हो जाएगा, and then the pollen tube will become turgid and will open when it comes in contact with that of synergid cells. So it is, as it is broken or it is burst open with the help of the great force of entire cytoplasm present in pollen tube, the male gametes are injected forcefully into, कहाँ पे जाएंगे, एम्ब्रियो सैक में. इनको क्या कर दिया जाएगा, भेज दिया जाएगा. So see here, try to see over here, there are 2 male gametes available. As soon as this pollen tube will rupture, the male gametes will enter in with the help of a great force. So this type of transfer of male gametes up to the ovule is called as siphonogamy. Remember this word, siphonogamy. What siphon is tube. आपको साइफनिंग वर्ड पता होगा, खास करके जिन लोगों के पास फिश टैंक होगा उनको तो ये 100% पता होगा, है ना। फिश टैंक पूरा भरा होगा तो उसको साफ करने के लिए ट्यूब यूज करते हैं। ट्यूब के अंदर पानी भरेंगे और एक एंड अंदर डालेंगे, एक एंड बाहर, और फिर फिश टैंक का पानी हम लोग बाहर खींच के ले सकते हैं। दैट इज साइफोनोगमी, ठीक है। तो उसको साइफनिंग कहते हैं। यहाँ पे इसी तरह से ट्यूब यूज किया हुआ है, पोलन ट्यूब यूज किया हुआ है। दैट पोलन ट्यूब रीचेस अप टू ओव्यूल एंड सेंड्स pollen grains. Is it? No. Now we should say male gametes in embryo sac. So it is called as siphonogamy. So this word is very important. So fusion of one of the haploid male gamete will be done now with egg, which is also haploid, and the other male gamete will fuse with that of secondary nucleus. Remember, secondary nucleus is diploid in nature. Okay. So this is called as fertilization occurring 2 times, so called as double fertilization. अभी syngamy क्या होता है? Syngamy actually is a fusion of one of the male gametes or one of the haploid male gametes with that of haploid egg cell so as to form diploid zygote. This event is called as syngamy. It is also called as generative fertilization because this fertilization will develop into zygote and zygote will develop into next generation individual. Okay. Zygote will be developed into embryo. Embryo will be capable of developing into a new sporophyte. Then triple fusion is next event. Why triple fusion? Triple का मतलब होता है 3 times. Okay. There is a secondary nucleus present in embryo sac. This is secondary nucleus. Secondary nucleus, find it well. It is diploid. Okay. Just try to recover whatever we studied last time. One nucleus from chalazal end, one nucleus from micropylar end come at the center and fuse. So each nucleus coming from chalazal end and micropylar end are haploid in nature. These nuclei will fuse so as to form diploid nucleus. This diploid nucleus is called as, क्या कहेंगे उसको हम लोग? Secondary nucleus. This secondary nucleus is diploid. So one of the male gametes, as we said, will form zygote. Other male gamete will go deep into it and will fuse with this diploid nucleus or secondary nucleus so as to form tertiary nucleus. हाँ, secondary से tertiary बनेगा. Secondary का मतलब diploid था. अभी एक और male gamete उसके अंदर fuse होने के बाद ये tertiary बन जाएगा. So remember this very well. It becomes tertiary nucleus. This tertiary nucleus is also called as primary endosperm nucleus. Why primary endosperm nucleus? Because this nucleus will divide and redivide so as to form endosperm. What is endosperm? Endosperm is a nutritive tissue for development of embryo. ठीक है, अभी यहाँ पे कुछ हम लोग को थोड़ा सा नीट पॉइंट ऑफ व्यू से डिस्कस करना चाहिए. Okay, remember very well, whenever a kind of ovule is developed or matured, matured ovule has got the antipodal cells on chalazal end, which are all haploid in nature. Okay, towards micropylar end, there are 3 cells, 2 synergid cells and 1 egg cell together forming egg apparatus. All are haploid in nature. Middle, there is a secondary nucleus present. Secondary nucleus is diploid in nature. So try to remember these things very well. And after fertilization होने के बाद क्या हो जाएगा? Antipodal cells will remain as it is. Synergid cells will degenerate. Antipodal cells भी degenerate ही हो जाएंगे बाद में, but उनके साथ कुछ हादसा नहीं होता है. There is no event occurring with that of antipodal cells. Synergid cells कम से कम पानी provide करते हैं to pollen tube so as to become turgid and burst open. So along with pollen tube, even the synergids might be breaking off. ठीक है. Zygote that is formed is diploid because zygote is formed by fusion of one haploid male gamete and one haploid female gamete. So zygote is diploid in nature. Okay, and primary endosperm nucleus is triploid in nature. Primary endosperm nucleus is triploid in nature. Triploid का मतलब समझ गए आपको? हाँ, so remember this very well. कुछ questions format करेंगे इसके ऊपर. देखो, in case of angiosperm, male, very carefully you have to listen and you have to answer this question in a chat box, whatever I'm asking. Okay, listen very carefully. Male body or male plant is having say 8 chromosomes. Female plant will be having 6 chromosomes in a diploid cell. They fertilize. They fertilize. Okay. So pollen grain from this male plant having 8 chromosomes was shredded onto the stigma of a female plant having 6 chromosomes. Then there was a fusion, double fertilization carried out. How many number of chromosomes will be there in primary endosperm nucleus? Got it well? What I asked? My question is very simple. How many chromosomes will be there in primary endosperm nucleus? PEN means primary endosperm nucleus or tertiary nucleus. ठीक है, इसमें कितने क्रोमोसोम्स होंगे? तुम लोग चैट बॉक्स में उसका नंबर डाल सकते हो. I am once again repeating, a male plant having 8 chromosomes in a diploid cell, female plant having 6 chromosomes in a diploid cell. There was a fertilization between these two plants, as a result of which primary endosperm nucleus was developed. Question is, how many chromosomes will be there in primary endosperm nucleus? This is a NEET level question. तो देखो, अभी यहाँ पे आपको सब कुछ याद रखना पड़ेगा जो मैंने थोड़ी देर पहले एक्सप्लेन किया था कि कौन सा न्यूक्लियस में कितना कितना प्लॉइड ही रहता है. सो एंटीपोडल सेल्स आर हैप्लॉइड, है ना. देन egg cell is haploid, synergid cells are haploid, but secondary nucleus is diploid. This is before fertilization. After fertilization, synergid cells are haploid, antibodal cells are haploid, zygote formed is diploid, and primary endosperm formed is triploid. So, with the help of this particular information, you have to answer my question. अरे कोई तो ट्राई करो. Yes, anyone? (46) अनुभूति, how it will be 46? ओके, और कोई देना चाहेगा, आंसर देना चाहेगा? (48) 28. See, you have not understood the basic of double fertilization. आपको नहीं समझा. Okay, the answer is 10. कैसे? Let us explain. Let us understand. Okay, fine. So, मैंने क्या कहा था? मेल के अंदर कितने क्रोमोसोम है? डिप्लॉयड मेल बॉडी हैज गॉट 8 chromosomes. So, haploid male cell will be having 4 chromosomes. Am I right? Just put yes or no in the chat box. I said diploid male has 8 chromosomes. So, haploid male cell will be having 4 chromosomes. Is it right or not? Fine. Good. Okay. Then I said female plant, diploid female plant has 6 chromosomes. So how many chromosomes will be there in haploid female cell? Put the number. 6 is diploid, so 3 will be haploid. Very nice. Good. Okay. यहाँ तक सबको समझ में आया? बराबर है. Now next question. Next question. Secondary nucleus कैसे बनता है? One haploid nucleus from chalazal end and one haploid nucleus from micropylar end will fuse together so as to form secondary nucleus, which is diploid. So how many chromosomes will be there in secondary nucleus? How many chromosomes will be there in secondary nucleus? Simple question. One haploid female cell from chalazal end carrying 3 chromosomes and other from micropylar end fuse together so as to form secondary nucleus. So how many chromosomes will be there in secondary nucleus? Secondary nucleus is diploid. अरे नहीं समझ में आया? देखो ये तीनों क्रोमोसोम्स, ये तीनों सेल्स हैप्लॉइड है. 5 कैसे हुआ? 6. इट हैज टू बी. यस, जैतून सेड करेक्ट. सो यहाँ से जो सेल आने वाला है, चलाजल एंड से जो सेल आने वाला है, हैप्लॉइड सेल, इट हैज गॉट 3 क्रोमोसोम्स. और माइक्रोपाइलर एंड से जो सेल आने वाला है उसमें भी है 3 क्रोमोसोम्स. whenever they fuse to form secondary nucleus there will be 6 chromosomes. यहाँ तक समझ में आया? Fine. Now male gamete that is coming through pollen tube, how many chromosomes will be there in each male gamete? हम लोगों ने कहा था diploid male plant is having 8 chromosomes. Very good. 4 chromosomes will be there in male gamete. Each male gamete will have 4 chromosomes. Now see what is happening. One male gamete is fusing here with zygote. तो zygote में कितने chromosomes होंगे? zygote में कितने chromosomes होंगे? उसमें एक फीमेल न्यूक्लियस है, एक मेल न्यूक्लियस है. फीमेल न्यूक्लियस will have dash number of chromosomes in our case study that we took into consideration, and male will be having dash number of chromosomes. So how many chromosomes will be there in zygote nucleus? 4 कैसे होगा? It will not be 4. 4 is wrong answer. Male gamete is having 4 and female gamete is having 3. So when the zygote is formed, how many chromosomes will be there? Yes, 7 chromosomes will be there. Fine. अब चलते हैं इसके पास. This is endosperm, primary endosperm nucleus. Primary endosperm nucleus fertilization से पहले secondary nucleus था. Secondary nucleus was having 6 chromosomes, right? After fertilization with one male gamete, how many chromosomes will be there here? After fertilization. Before fertilization it has got 6, but after fertilization how many will be there? Vivek, you are saying before fertilization. Fertilization हुआ है अभी. Male gamete उसके से, ये देखो ये male gamete यहाँ पे आया है. ये दो female gamete already थे. Each female gamete was having 3 and each male gamete will have 4. So how many total number of chromosomes will be there in? Yes. कैसे समझ में आया सबको? अभी 10 आंसर कैसे है? हाँ, ओके. सो इट्स वेरी सिंपल. जितना क्रोमोसोम नंबर फीमेल के पास है उतना का उतना ही रखने का, और जितना क्रोमोसोम नंबर डिप्लॉयड मेल के पास है उसका हाफ करने का, उसका हाफ करने का. ठीक है, दैट विल गिव यू. यस जिज्ञासा, यू आर राइट, उसका हाफ करने का. तो दैट विल गिव यू. अभी देख, और एक सवाल पूछता हूं, उसका मुझे सही जवाब चाहिए. मैं अभी कुछ भी एक्सप्लेनेशन नहीं दूंगा. ठीक है. मेल प्लांट हैविंग 20 chromosomes fertilizes a female plant having 18 chromosomes. Listen very carefully. Male plant having 10 chromosomes in a diploid cell fertilizes a female plant having 18 chromosomes in a diploid cell. How many chromosomes will be there in? Very carefully listen questions. How many chromosomes will be there in secondary nucleus, primary endosperm nucleus and zygote? मैंने अभी 3 के लिए पूछा है, तो तीनों को जवाब दे दो. फर्स्ट वाला फर्स्ट, सेकंड वाला सेकंड, थर्ड वाला थर्ड. मैंने ऑर्डर क्या रखा है? सेकेंडरी न्यूक्लियस में कितने होंगे क्रोमोसोम्स, देन एंडोस्पर्म न्यूक्लियस में कितने होंगे, प्राइमरी एंडोस्पर्म न्यूक्लियस में कितने होंगे, और जाइगोट में कितने होंगे. मेल इज हैविंग 20 chromosomes in a diploid cell. Female is having 18 chromosomes in a diploid cell. How many chromosomes will be there in primary endosperm nucleus? Put that number first. Then, sorry, first question मैंने पूछा था how many chromosomes will be there in secondary nucleus, then primary endosperm nucleus and zygote. Or if I would like to make a question more complicated, I will ask which of these 3 will be having same number of chromosomes. Which of these 3 विल बी हैविंग सेम नंबर ऑफ क्रोमोसोम्स, और फिर मैं उसके ऑप्शन बनाऊंगा. सेकेंडरी न्यूक्लियस, प्राइमरी एंडोस्पर्म न्यूक्लियस. सेकेंडरी न्यूक्लियस zygote, zygote primary endosperm nucleus, and primary endosperm nucleus with that of secondary nucleus. अरे इतना एक्सप्लेन करने के बाद, ये इतना सिंपल सवाल है, आप लोग आंसर नहीं दे पा रहे हो. सो हाउ यू आर गोइंग टू डू इट इन नीट? ये सब सवाल नीट में पूछा जाएगा, एंड इन वन सिंपल minute you have to answer this. Secondary nucleus will be having 18. Okay, fine. टर्शियरी न्यूक्लियस में कितना रहेगा? 28. जाइगोट में कितना रहेगा? जाइगोट में कितना? 19. दैट्स राइट. कीर्तना, गुड अटेम्प्ट. ₹1 बख्शिश तुम्हें बनता है. In a physical lecture, I would have given you ₹1 बख्शिश for giving this nice answer, but you took much time. It should not be. अभी कैसे सबको मालूम पड़ गया? सी, जितने नंबर ऑफ क्रोमोसोम्स फीमेल प्लांट में बताए गए हैं उतने ही सेकेंडरी में रहेंगे, उसके हाफ एंटीपोडल्स में रहेंगे. ठीक है, डिप्लॉइड के हाफ हैप्लॉइड ना, सो एंटीपोडल सेल्स आर हैप्लॉइड, synergid cells are haploid, then egg cell is haploid. So all of them, they will be having half of the chromosomes of diploid female cell. ठीक है, secondary nucleus will be having number of chromosomes equal to diploid female cell. Male gamete will be having half number of chromosomes with respect to the diploid male cell. ठीक है, and then you have to just add up, taking into consideration zygote कैसे बनता है. Zygote is made from haploid male gamete and haploid female gamete. तो half of male gamete plus half of female gamete, or half of male chromosome in diploid cell plus half of female chromosome in diploid cell will give you number of chromosomes in zygote. ठीक है, yes. Then triploid nucleus or endosperm nucleus, diploid of female gamete or female diploid of female cell plus half of male cell chromosomes will be there in primary endosperm nucleus, और सेकेंडरी में जितना फीमेल में है उतना ही रहेगा. सो इस तरह के क्वेश्चंस आएंगे, सो यू हैव टू सॉल्व इट. अभी तो मैंने सिंपल कैलकुलेशन करने के जैसा नंबर दिया था, कभी कभी ये कॉम्प्लेक्स होंगे. सो देखो, ये इस तरह के हायर लेवल के क्वेश्चंस. तो मैंने एक सवाल और पूछा, which of the two following will be having same number of chromosomes, और उसके लिए हम लोगों ने ऑप्शन बनाए, जैसे कि सेकेंडरी न्यूक्लियस, एंडोस्पर्म न्यूक्लियस एक ऑप्शन, सेकेंडरी न्यूक्लियस जाइगोट दूसरा ऑप्शन, टर्शियरी न्यूक्लियस और जाइगोट और टर्शियरी न्यूक्लियस एंड एंडोस्पर्म किसमें सेम रहेंगे? Number of chromosomes in zygote and number of chromosomes in secondary nucleus will be same. Is it? No. एक ऑप्शन आना चाहिए, none of this. Okay? क्योंकि मेल और फीमेल में ऑड नंबर है. अगर मेल और फीमेल में इवन नंबर ऑफ क्रोमोसोम्स होंगे, तब उस हालत में नंबर ऑफ क्रोमोसोम्स इन सेकेंडरी न्यूक्लियस एंड नंबर ऑफ क्रोमोसोम्स इन जाइगोट विल बी सेम. ठीक है, नीट करेंगे तभी और डिस्कस करेंगे डिटेल में. will not waste time over here right now. जितना बताया उतना याद रखो बराबर से and try to do it. Okay, so let us carry our story forward. Triple fusion आपको समझ में आ रहेगा. So triple fusion will give primary endosperm nucleus. Primary endosperm nucleus will develop into triploid endosperm tissue. This endosperm tissue will be a food for developing embryo. Fine. So our story will have to consider significance of double fertilization. Significance का मतलब क्या फायदा है ये करके, ट्रिपल फ्यूजन करके क्या फायदा होगा, या फिर डबल फर्टिलाइजेशन करके क्या फायदा होगा, इसी को हम लोग कहेंगे पोस्ट फर्टिलाइजेशन चेंजेस. ओके, सो दिस इज द एंटायर साइकिल वी आर शोइंग ओवर हियर. सो यहां से हम लोग शुरू करते हैं स्पोरोफाइट. develops flower. Flower has got female part, a male part. Female part ovule develops into female gametophyte. Female gametophyte will be there, matured. Then these are microsporangia, produce microspores, pollination, fertilization, and again over here zygote is produced. Egg cell will get converted into zygote. Zygote will develop into double fertilization के बाद it will develop into a seed. This will be called a seed. Seed will germinate once again so as to give new sporophyte. So double fertilization के फायदे क्या है? Development of fruit and seed will start. Then there will be haploid egg will become diploid zygote and that turns into embryo. That is another characteristic feature. Okay. Then diploid condition will be restored. सब कुछ समझ में आ रहा है ना, इजी है, इसमें ज्यादा कुछ डिफिकल्ट है नहीं. पहला क्या बोला हम लोगों ने? डेवलपमेंट ऑफ फ्रूट एंड सीड विल स्टार्ट. डेवलपमेंट स्टार्ट हो जाएगी. ओके. सो zygote or ovule when gets matured, developed completely, it will become seed, and the ovary when gets matured completely will develop into fruit. So fruit is defined as fertilized and matured ovary or ripened ovary. Fertilized and ripened ovary will be called as fruit. Seed will be fertilized and developed ovule. So these two will be there. Haploid egg का diploid egg बन रहा है या diploid zygote बन रहा है, means once again we can say the diploid condition is restored. Okay, sporophyte was diploid. It develops haploid gametes and after it will become once again fertilization, it will become diploid again. Ovule will get transformed into seed. Ovule का conversion seed में हो जाएगा. Ovary will now start accumulating food material. The wall of ovary is called as pericarp. This pericarp is thin before fertilization. After fertilization, it will start undergoing changes. It will start accumulating food. It will become thick. The wall will expand little bit and will undergo ripening so as to form fruit. Okay, there are different types of fruits available which we have studied last year. So we'll not get into it once again. Integuments which are present on the ovule. Integuments आपको याद होंगे, coverings around the ovule. They will be converted into seed coats or testine tegmen. So these all changes are essential. ये एक 2 मार्क का क्वेश्चन बनता है. Give significance of double fertilization or post fertilization changes. Endosperm, remember, endosperm is a food for embryo. So that is developed during this particular process. So primary endosperm nucleus is formed that will produce endosperm tissue, or this endosperm tissue we can say is a nutritive tissue for the developing embryo. These are the characteristic features over here. And it will avoid polyspermy. Polyspermy का मतलब क्या? एक ही ओव्यूल में एक से ज्यादा क्या कहते हैं स्पर्म्स, मतलब मेल गैमेट्स नहीं आने चाहिए. That will be avoided. So now we have to go for development of endosperm. Development of endosperm का मतलब क्या होगा कि endosperm किस तरह से डेवलप होने वाला है, that we have to understand. Now after fertilization, जब भी fertilization हो जाएगा तो instantaneously, immediately certain changes start occurring in the ovule and that will lead to formation of seed and formation of endosperm. पहले endosperm बनना चाहिए because before developing embryo, the food for embryo must be there ready. Okay, so जैसे ही fertilization हो जाता है तो diploid zygote and triploid primary endosperm will start developing simultaneously, but predominantly developing is endosperm. Okay, other cells of embryo sac will get disorganized, मतलब ये मर जाएंगे या फिर disintegrate हो जाएंगे, उसका कुछ महत्व नहीं रहा. What are the other cells now? Synergid cells and antipodal cells. उसका कुछ फायदा नहीं है, so they will degenerate. Primary endosperm nucleus will start dividing and redividing mitotically so as to form nutritive tissue. This nutritive tissue will be considered as endosperm tissue, which is a food for embryo. There are 3 different ways of developing endosperm tissue. Those 3 we have to consider. First is called as nuclear type of endosperm. What does it mean, nuclear type of endosperm? The triple nucleus or triploid nucleus will start dividing and redividing rapidly by the process of mitosis so as to form several nuclei. सिर्फ nuclei बनते जाएंगे. Central vacuole will be developed in the center of the ovule now, or center of the ovule part. This will push all the nuclei towards periphery. So न्यूक्लियस जैसे बनते जाएंगे एक वैक्यूल डेवलप हो जाएगा. दिस वैक्यूल विल send a nuclei towards the periphery, as a result of which you could see the nuclei all over the peripheral region. The cell wall formation will take place to form multicellular type of endosperm. The cell wall formation will be incomplete, as in the case of coconut, sunflower, wheat, etc. Okay, so यहाँ पे जो मेंशन किया हुआ है, ट्राई टू अंडरस्टैंड दिस. कौन सा एग्जांपल दिया है? कोकोनट का. आई वुड लाइक टू टेक दैट एग्जांपल. ओके, so coconut has got multicellular type of endosperm as the periphery, at the periphery, and the free nuclear type of vacuolated endosperm at the center. कोकोनट आपको मालूम होगा, है ना? नारियल, नारियल के अंदर खोपरा होता है और पानी भी होता है, मलाई भी होता है, पानी भी होता है. बोथ आर एंडोस्पर्म. ओके, तो आपको किसने पूछा कि व्हाट इज लिक्विड एंडोस्पर्म? तो लिक्विड एंडोस्पर्म का बेस्ट एग्जांपल है नारियल, कोकोनट. ओके, इवन सुपारी, उसमें भी सिमिलर टाइप है. और ताड़गोल आपको पता होगा, तो भी वो भी सिमिलर है. ठीक है, दिस थ्री एग्जांपल्स यू हैव टू रिमेंबर, दे आर हैविंग लिक्विड एंडोस्पर्म प्रेजेंट इन देम. फाइन, फेमस एग्जांपल्स ऑफ कोर्स. अभी नारियल का पानी जो है वो एंडोस्पर्म है. फाइन, तो उसमें दोनों टाइप के डेवलपमेंट होते हैं. Nuclear type of development will be there, nuclear type of endosperm development will be there. So what will happen? Centrally a vacuole will develop. This vacuole after development will push the endosperm towards the periphery. इसका बनेगा खोपरा. ओके, इसको हम लोग कोकोनट का मलाई कहते हैं. अंदर कुछ पानी रहेगा, ये पानी जो है उसको हम लोग कहेंगे लिक्विड एंडोस्पर्म. सो दिस टू आर इंपॉर्टेंट. ओके, रिमेंबर इट वेरी वेल. देन it is found in 161 angiospermic families, liquid endosperm. तो nuclear type of development कैसे होता है, that we have to understand. Say it is like this. Okay, single triple nucleus is there. Triple nucleus क्यों कहेंगे? Because it is a triploid nucleus. This triple nucleus will start dividing and redividing. See, this is a vacuole developing. Vacuole will make all the nuclei to get arranged on the periphery. So it is a free nuclei present over there, and that's what is the endosperm available. Fine. First type of endosperm is nuclear type. Okay. Second type of nucleus, nucleole. Second type of nucleus, or what we can say endosperm, will be cellular type. अभी cellular type में क्या हो जाएगा? The first triple nucleus will be there. It will keep on dividing and redividing mitotically so as to form many nuclei. And whenever nuclei develop, nuclear division will be followed by cell wall development or cell wall formation. तो nuclei के बीच में cell wall तैयार हो जाएगी. So all the cells will become separate with, or all the nuclei will get separated from each other in an individual cell. Around 72 families like balsam, then petunia, adoxa, etc. They will be having this type of development. So what is cellular type that we are discussing over here? Nucleus divides into two nuclei, cell wall formation. And whenever there is a nuclear division taking place, always a cell wall will be formed. So this entire endosperm will be full of cells. That will be called as cellular type of endosperm. Third is called as halobial. Halobial is partially nuclear and partially cellular. First nuclear division will be followed by formation of a transverse cell wall. बराबर से सुनो, ध्यान से सुनो. न्यूक्लियर में क्या हुआ? न्यूक्लियर एंडोस्पर्मिक डेवलपमेंट में न्यूक्लियस कीप ऑन डिवाइडिंग एंड रीडिवाइडिंग. वैक्यूल डेवलप्ड एंड न्यूक्लियस वर टेकन टू द पेरिफेरी. सेल में क्या हुआ? सेलुलर टाइप में क्या हुआ? न्यूक्लियर डिवीजन वाज फॉलोड बाय सेलुलर डिवीजन इमीडिएटली और सेल वॉल फॉर्मेशन. यहाँ पे क्या हो रहा है? हेलोबियल में क्या हो रहा है? फर्स्ट न्यूक्लियर डिवीजन विल फॉर्म फर्स्ट सेल वॉल फॉर्मेशन और ट्रांसवर्स सेल वॉल फॉर्मेशन, एंड देन इन ईच ऑफ द सेल, दो सेल बन गए ना, हर एक सेल में एक एक न्यूक्लियस है. वन सेल इज प्रेजेंट टुवर्ड्स चलाजल एंड, अदर इज प्रेजेंट टुवर्ड्स माइक्रोपाइलर एंड, एंड नाउ इन ईच ऑफ दिस व्हाट विल हैपन? द न्यूक्लियर डिवीजन विल बी continued n number of times. There will be a vacuole developed and the nuclei would be sent to periphery. Okay, so cell wall will develop in between the nuclei of the micropilar end, but it will not be there in other end. So this type of endosperm will be intermediate between free nuclear and the cellular type. दोनों के बीच का ये characteristic feature है. found in helobials like एस्पोडिलस. तो ये एक एग्जांपल इसका याद रखना बराबर से कैसे होता है. वी आर गोइंग टू अंडरस्टैंड ओवर हियर क्या होता है इसके अंदर. सी दिस इज हाउ इट विल डेवलप. So endosperm nucleus, primary endosperm nucleus divides into two. Then there will be formation of nucleus towards micropylar end to end towards chalazal end. Okay. So what is happening towards chalazal end? Chalazal end जो है वहाँ पे cell division means nuclear division होके cell wall बनता है, but micropylar end में ऐसा नहीं होता है. So it is having both the types available or both the types available. Fine. That's what is called as. Okay. Now endosperm क्या करता है? Endosperm tissue when develops, it will try to absorb or will try to utilize nucellar cells, remaining nucellar cells. भी आप क्या बोल रहे हो समझ में नहीं आ रहा है सर. ये न्यूसेलस कहाँ से आया, न्यूसेलर सेल्स कहाँ से आया. तो उसके लिए ट्राई टू रिकॉल वॉट एवर वी डिस्कस लास्ट टाइम. डेवलपमेंट ऑफ फीमेल गैमेटोवाइट के हम लोग ने वक्त हम लोगों ने कहा था ओव्यूल inside it has got a mass of parenchymatous cells. This mass of parenchymatous cells is called as nucellus. One of the nucellar cells developing fastest and largest will become megaspore mother cell, जिससे बनता है embryo sac. Embryo sac बढ़ेगा, but it will not make utilization of all the nucellar cells. The cells of the nucellus will remain. Those cells of the nucellus will be utilized by endosperm. So cells of nucellus will be used by endosperm, means nucellar cells will be converted into food for embryo. ये एक चीज याद रखना. Okay. Now development of embryo कैसे होगा? That we have to understand. This is again a multi-step process. So development of embryo will be called as embryogenesis. This embryogenesis will take place at micropylar end and immediately after the formation of small amount of endosperm. मैंने क्या कहा था कि endosperm tissue will proceed further और endosperm tissue will go ahead. पहले endosperm tissue तैयार होगा, उसके बाद में zygote का डेवलपमेंट शुरू होगा. ओके क्योंकि थोड़ा तो फूड चाहिए उसको. Nucellus is not a food which can be utilized by embryo, so it requires some digested type of food, and that's how development of endosperm is must. So zygote cell which was developed will divide so as to form proembryo. This proembryo is a two-celled structure. Proembryo क्या है? दो cell structure है. Now try to understand this. This is a egg cell, fertilized egg cell, which will divide into two cells. So this will be called as proembryo, having two cells. One cell is called as apical cell, and other cell is called as basal cell. So there is a large basal cell available, and there is a small apical cell available. Basal cell is also considered as suspensor initial cell or suspensor initiating cell, and this is generally formed towards micropylar end. मतलब जो zygote बन गया है, zygote का first division होगा so as to form two unequal cells. One larger cell towards micropylar end will be called as basal cell, and other smaller cell towards chalazal end will be called as apical cell. Okay, this basal cell available over here will further keep on dividing and redividing so as to form a small chain of cells which is called as suspensor. Smaller terminal cell or apical cell or embryonal initial cell will be formed towards chalaza. It will keep on dividing and redividing. Okay. Now what happens to basal cell? It will start dividing and redividing so as to form postorial cell or suspensal cell. वापस दो में बन जाएगा ये. So postorial cell मतलब क्या? जो होस्ट करेगा, पकड़ के रखेगा. और suspensor cell का मतलब क्या? एक चेन बनाएगा ये. So suspensor cell will divide so as to form 8 to 10 cell structure, which is a small chain like structure. This structure will be called as suspensor. Suspensor क्यों? Because it is going to suspend the embryo close to endosperm. आगे के डायग्राम में ये दिखेगा आपने को. So we will be capable of watching it. Now see here, this is a basal cell, or this was a basal cell which has got converted now into host cell, and there was a suspensor cell. suspensor cell divided so as to form this long chain like structure. This will be called as suspensor. आगे embryo है. This embryo will be brought close to endosperm. Okay. So last cell of suspensor is called as hypophysis, that during the development forms part of radicle and root cap. ये जो last cell है इसका बनेगा radical and root cap. Okay. So that is a characteristic feature. Suspensor का काम क्या रहेगा? Suspensor will bring embryo very close to endosperm, or it will push the embryo deep into the endosperm. Endosperm, remember, is a food for developing embryo. Embryonal initial cell, जो यहाँ पे था embryonal initiation cell, will start dividing into certain cells. There will be 3 systematic divisions available so as to form 8 cell structure. अभी यहाँ पे आप लोग को 4 cells दिख रहे हैं, but उसको octane कहा है हम लोगों ने. Octane is a group of 8 cells. So 4 cells ahead, 4 cells behind. Okay, that's how. One division will form 2 cells. 2 divisions will form 4 cells. And 3 divisions will form 8 cells. Okay, number of cells produced will be equal to 2 raised to the number of divisions. So 5 division हुए तो 2 raised to 5 means 32 cells will be developed. Okay, so likewise it will be. The cells will be arranged in 2 rows of 4 each. I told you. आगे का रो 4 cells का, पीछे का रो 4 cells का. So octane is produced. This octane will be then start developing in a definite fashion. Okay, it will start dividing and redividing faster so as to form sometimes a lower layer that produces hypocotyle and radical, and it will give a triangular structure of embryo. This triangular structure will have developing speed more on the ends. इस end के ऊपर और इसके end के ऊपर बहुत speed है. That's how, see, the growth will be here more and growth here also will be more. तो दोनों तरफ growth ज़्यादा हो जाएगा. So lower cell जो है वो produce क्या करेगा? Hypocotyle and radical, whereas upper layer will produce terminal plumule and dividing further so as to form heart-shaped cotyledons. So these are the cotyledons that are made initially. So this will be a chordate-shaped embryo. Chordate का मतलब heart-shaped embryo. Okay. So this chordate-shaped embryo will keep on dividing further. ये था पुराना chordate shape जो था, उसको देखो अभी division बहुत ज़्यादा हो गया. So it will form this bifurcated structure. This bifurcated structure will be responsible for formation of horseshoe-shaped nucleus or horseshoe-shaped embryo. तो देखो ये पूरा का पूरा टर्न हो जाएगा and it will form 2 cotyledons. will be clear now. एक और 2 cotyledons बराबर हो गए. यहाँ पे जो बेस है इससे रेडिकल और प्लूमल बनने वाला था और यहाँ पे जो है उसमें हाइपोकोटाइल बनने वाला था. ठीक है. So in case of monocots also the development will be just similar up to octane stage, but further it will get diversified. Here the single cotyledon will be produced at the terminal end and plumule will be produced on lateral side. Plumule and radical of course will be produced. The single cotyledon is called as scutellum. Monocots में जो single cotyledon होता है उसको scutellum कहा जाता है. So try to remember this very well. Then protective cover will be there found over the plumule. It is called as coleoptile, and protective covering present around the radical will be called as coleorhiza. देखो ये words बराबर याद रखना. MCQs में ये words हमेशा पूछे जाएंगे. Protective covering around plumule will be coleoptile. Plumule में P है, coleoptile में P है. ठीक है. Radical का covering इसको कहेंगे कोलियोराइजा. रेडिकल में R है और कोलियोराइजा में R है. ठीक है. कुछ इस तरह से आइडियाज डेवलप करके आपने को याद रखना पड़ेगा, अदरवाइज इट इज डिफिकल्ट. फाइन. सो देयर विल बी फॉर्मेशन ऑफ सच काइंड ऑफ थिंग. देन डेवलपमेंट ऑफ मोनोकॉट एम्ब्रियो वो कैसे होगा वो थोड़ा सा हम लोग डायग्रामेटिकली देख लेते हैं. आई टोल्ड यू अर्लियर डेवलपमेंट इज सिमिलर. वो स्पोर इज प्रोड्यूस्ड, सस्पेंसर सेल, मिडल सेल एंड टर्मिनल सेल. देन there will be suspensor produced and this octane will be produced. Octane तैयार होने के बाद there will be one single cotyledon produced. There will be a plumule available. Lateral side में कहा था हम लोगों ने plumule है, base में radical है, और ये पूरा कोटिलिडॉन है और यहाँ पे हाइपोकोटाइल है. Okay, so this radical cotyledon and plumule will be available, and a sectional view it will look like this. Okay, so this is development in monocots. Now we have to consider seed and fruit development. Okay, कैसे होगा? Pollination will be followed by fertilization, which leads to development of seed and fruit. तो उसमें क्या क्या changes होते हैं उतने से अपने को याद रखना है. Seed will germinate so as to give the offspring. Okay, getting it? The integuments that are available, they will form seed coats. Seed की जो coverings है वो बन जाते हैं. देखो पहले वाले क्या था? न्यूसेलस. न्यूसेलस में से एक सेल बना, मेगास्पोर मदर सेल्स. Rest of the cells remained as it is, but nuclear cells will be utilized by endosperm nucleus, और endosperm tissue बनेगा. Endosperm tissue will be utilized by embryo so as to form cotyledons. So cotyledon is a reserved food for embryo. ठीक है, ये एक मतलब न्यूसेलर सेल्स का क्या हुआ आपको समझ में आया. ठीक है, देन एग सेल बना था. एग सेल विल डिवाइड एंड रीडिवाइड सो एज टू फॉर्म एम्ब्रियो, तो उसका एंड हो गया. देन देयर वर टू coverings present over. Those coverings were called as earlier integuments. Now they will become seed coats or seed coverings. Okay, there are two coverings available. Outer will be called as testa and inner is called as tegmen. So testa and tegmen are the two coverings available. Testa being outer will be naturally hard, rough and tough, and inner tegmen will be soft and thin, delicate. Okay, न्यूसेलस के बारे में क्या होता है मैंने बताया तुम लोगों को, वही यहाँ पे दिखा हुआ है. न्यूसेलस इज यूज्ड एज ए फूड बाय डेवलपिंग एंडोस्पर्म एंड इट मे परसिस्ट इन सम केसेस एज पेरिस्पर्म. हाँ, अभी ये इंपॉर्टेंट है, ये वर्ड नया आया यहाँ पे, पेरिस्पर्म. हम लोगों ने क्या कहा पहले कि न्यूसेलस विल बी यूटिलाइज्ड बाय एंडोस्पर्म और न्यूसेलस विल बी कन्वर्टेड इनटू एंडोस्पर्म टिश्यू. लेकिन किसी किसी फ्रूट्स में, किसी किसी सीड्स में ये पूरी तरह से यूटिलाइज नहीं हो जाता. एंडोस्पर्म विल नॉट यूटिलाइज एंटायर न्यूसेलस. स्मॉल थिन लेयर ऑफ एंडोस्पर्म, स्मॉल थिन लेयर ऑफ न्यूसेलर सेल्स विल रिमेन एंड देन इट विल बिकम पेरिस्पर्म. ठीक है, क्योंकि वे पेरिस्पर्म, पेरी का मतलब पेरीफेरी और स्पर्म का मतलब क्या हुआ? एम्ब्रियो सैक, है ना, उसका कवरिंग बनता है. दैट्स व्हाट इज कॉल्ड एज पेरिस्पर्म. समटाइम्स endosperm also is not used completely and remains in seed. So if endosperm remains in seed as it is, it will be called as endospermic seed or albuminous seed. देखो बराबर से कितने छोटे छोटे पॉइंट्स है. शायद तुम लोग को एक पार्ट थोड़ा बोरिंग लगता रहेगा. ओके, बट दिस इज वेरी इंपॉर्टेंट फ्रॉम नीट पॉइंट ऑफ व्यू. Remaining part of nucellus, if it is not utilized completely by endosperm, will be called as perisperm. Okay, if embryo is not using endosperm completely, endosperm is remaining, then it will be called as endospermic seed or albuminous seed. Examples उसके दिए हुए castor, coconut, maize. ठीक है, so इसको हम लोग कहेंगे endospermic seed. If it is utilized completely, endosperm is utilized completely and converted into cotyledons, then it will be called as non-endospermic seed or exalbuminous seeds. ओके, सो पी, बीन एक्सेट्रा ये जो है उसमें सब में क्या है? दीज आर ऑल नॉन एंडोस्पर्मिक सीड्स और एक्सएल्बुमिनस सीड्स. ओके, अल्बुमिन का मतलब एंडोस्पर्म उनमें नहीं है. ओके, cotyledons may have stored food or may act as a photosynthetic. क्या बनेगा ये? स्टोर्ड फूड का काम करेगा या इट विल बिकम फोटोसिंथेटिक ऑर्गन इमीडिएटली आफ्टर जर्मिनेशन. जर्मिनेशन के बाद क्या होता है ये थोड़ा सा इंपॉर्टेंट है. किसी किसी सीड्स में ये फूड प्रोवाइड करेगा कुछ दिनों तक, या किसी किसी सीड्स में क्या हो जाएगा, ये बाहर आ जाएगा सीड से. ओके, तो जर्मिनेशन आर ऑफ डिफरेंट टाइप. हाइपोजियल जर्मिनेशन, एपिजियल जर्मिनेशन. In case of epigial germination, the cotyledons will come out along with the plumule, and in the initial stages, it will help in the process of photosynthesis. Okay, so that is one more characteristic feature to be remembered. Micropyle will remain as it is, as a small aperture for taking in water and oxygen during the dormancy period of the seed and during the germination of seed. Okay, So it will remain as a small pore over there, micropyle. Simultaneously, the wall of ovary will start developing. I told you, wall of ovary is called as pericarp. This pericarp will start developing. The wall will start becoming thick. It will start accumulating food material. As it accumulates food material, it will become thick. It will grow in size. Okay. Ultimately, it will get ripened so as to form fruit. तो सब राइप हो जाएगा. राइप मतलब समझ गए ना, पक जाएगा. वो फ्रूट कच्चा होता है, जैसे कैरी. कैरी आपको मालूम है, तो कच्चा कैरी कैसा लगेगा? खट्टा लगेगा. बट जब जब वो आम में कन्वर्ट हो जाएगा, मैंगो में कन्वर्ट हो जाएगा, it will taste good, will taste nice. So that's an idea behind it. Okay. So wall of ovary is called as pericarp. This pericarp, after complete development of a fruit, may be divided into three layers, like epicarp. The outer layer will be called as epicarp, middle layer will be called as mesocarp, and the innermost layer will be called as endocarp. Depending upon this epicarp, mesocarp, endocarp, the fruits may be further subdivided. ओके. जैसे कि कोई कोई फ्रूट्स होते हैं वो ड्राई फ्रूट्स होते हैं, कोई ड्राई डेहिसेंट फ्रूट होते हैं, कोई फ्रूट जो होते हैं वो नॉन ड्राई फ्रूट्स होते हैं. नॉन ड्राई मतलब फ्लेशी फ्रूट्स होते हैं. कुछ फ्रूट्स फाइबरस होते हैं, तो उनके अंदर फाइबर्स होंगे. सो ये सब जो डिविजन्स है फ्रूट्स के, दोज डिविजन्स डिपेंड ऑन द नेचर ऑफ आइदर एपिकार्प और मेसोकार्प और एंडोकार्प. ठीक है. नाउ सिग्निफिकेंस ऑफ सीड एंड फ्रूट फॉर्मेशन. क्या फायदा है सीड फॉर्म होके और फ्रूट फॉर्मेशन होके? क्या फायदा है? प्लांट्स को तो फायदा है, लेकिन अपने को भी फायदा है. सो दैट वी हैव टू अंडरस्टैंड. फ्रूट्स विल प्रोवाइड नरिशमेंट टू द डेवलपिंग सीड्स. सीड्स के अंदर जो डेवलपमेंट हो रहा है, उसके लिए फ्रूट्स जो है वो प्रोवाइड करेंगे नरिशमेंट. फ्रूट्स विल प्रोटेक्ट द सीड्स इन इमेच्योर कंडीशन. फ्रूट्स के अंदर सीड्स है, तो उनको बराबर से कवरिंग मिला है. whenever it is raw, unripe, it is very hard. कोई भी फ्रूट देखो, जब कच्चा होगा तो बहुत कड़क होगा, पकने लगेगा तो थोड़ा सा नरम पड़ सकता है. Depends. Okay. Seeds will serve as propagating organ, means dispersion of seed, dispersion of species. Fruits are attraction. So many animals which are living on trees, they will use these fruits as a food. They will eat the fruit and will disperse the seeds wherever they go. So that is what is given over here. Seeds are serving as propagating organs, मतलब अपने species को आगे बढ़ाने के लिए काम करते हैं. Seeds and fruits, they will develop special devices for dispersal of the seed. उसमें अलग अलग तरीके डेवलप हो जाएंगे. कभी कभी फ्रूट्स के ऊपर कांटे हो जाएंगे, कभी कभी सीड्स के ऊपर कांटे हो जाएंगे. सीड्स में भी स्टिकी इन सम केसेस, सीड्स में भी वेरी लाइट. Seeds may develop parachute-like structure, so they can be dispersed very easily along with the help of wind. So ऐसे बहुत सारे structures तैयार करेंगे so that there will be easy dispersal of the seed available. Structural or physiological adaptive mechanisms for survival of dormancy. Dormancy क्या होता है? Dormancy period is a period in which the seeds will remain as it is in that particular state only. They will not germinate. So from the formation of seed till the germination of seed, whatever the period is there, that period is called as period of dormancy. Okay. So seed is also a device where structural and physiological adaptations are must, so that it will remain dormant till the germination period. Okay. Dormancy will help to overcome the adverse environmental conditions. देखो अभी जनरल लाइफ साइकिल आप लोग देखोगे, अपने इंडिया में बारिश होती है सितंबर तक, हम लोग कहेंगे. सितंबर के एंड में फ्लावरिंग सीजन आता है. उसके बाद तुरंत इमीडिएटली फर्टिलाइजेशन होके सीड्स बन जाते हैं. एंड देन द वीड्स, जनरली विच वी कॉल देम एज वीड्स, uncultivated or non-cultivable plants, but which grow on the rain, onset of rain, will be called as weeds. So they will develop into seeds. Then after October, month of October, there will be a dry season. ठंडी का सीजन आ जाएगा, वो ड्राई होता है. तो जो फ्रूट्स है वो फ्रूट्स ड्राई हो जाते हैं, उनके सीड्स निकल पड़ते हैं बाहर, जमीन के ऊपर गिर जाते हैं. अभी यहाँ पे ड्राई कंडीशन है. उसके बाद आता है वापस से धूप, समर सीजन. समर में तो बहुत हॉट होता है. ओके. So during all this particular season, what should happen? The dormancy should be maintained properly, and the dormancy period should be overcome after beginning of rainy season, means normal condition आने के बाद वापस ये हो जाएगा. But till then the embryo should be well protected. Embryo का protection अच्छे से होना चाहिए, and that's how these seeds are developing thick covering around it. Mustard and viable seeds do not germinate in dormancy period. डॉर्मेंसी पीरियड में जर्मिनेट होंगे ही नहीं, एक पर्टिकुलर सीजन ही लगेगा उनको, तभी ये जर्मिनेट होंगे. During this period, there may be a dispersal of seeds, and the seeds may be carried out to the different areas that will help in development or spreading of species. Monocot seed and dicot seed में थोड़ा सा डिफरेंस है. क्या डिफरेंस है? मोनोकॉट विल बी हैविंग सिंगल कॉटिलिडॉन. ओके, ये एक ही कॉटिलिडॉन है, उसको स्कुटेलम हम लोग कहेंगे. सी देयर इज एपिकोटाइल, इज कवरिंग ऑफ इट. हाइपोकोटाइल, एपिकोटाइल क्या होगा? जो एक्सिस है उसका ऊपर वाला पार्ट है उसको एपिकोटाइल कहेंगे, नीचे वाला पार्ट जो है उसको हाइपोकोटाइल कहेंगे. देन देयर इज रेडिकल, कॉटिलिडॉन एंड सीड कोट विल बी अवेलेबल. This is embryo for monocot. This is embryo entirely for dicot. You could see there are two halves available of seed. There is only one half in the seed. There is endosperm available here in monocot. There is no endosperm persisting, but there are two cotyledons available. Okay, so these are the different structures that are developed. Now, some conditions are there apart from normal fertilization, but those conditions and rest of the things will try to keep it for our next time lecture. Okay, so